All right, got an exam on cell physiology coming up? Hey, don't sweat it. You are in exactly the right place. We're about to do a super focused review of all the must-know concepts. We're going to break it all down so you can walk into that test feeling totally prepared. Let's get into it.
Okay, let's just dive right in. We're starting with the big picture, right? Think of this as the map of your body's own internal ocean. And yeah, it seems kind of basic, but trust me, getting a handle on this distribution is the absolute foundation for everything. And I mean everything else we're going to cover.
So, here's our game plan. First, we're going to map out that internal ocean. Then we'll zoom way into the cellular frontier, the gatekeeper that controls, well, pretty much everything. From there, we'll get to the really cool part, the spark of life itself, those electrical signals that make our cells work. And finally, we'll wrap it all up with a lightning round of the key concepts you absolutely have to know to crush that exam.
First up, let's get our bearings. We're talking about the basic geography of your body's fluids. If you can nail this part, you're already on your way to mastering cell physiology. So let's lock it in.
Okay, this slide is a big one, so pay close attention here. For an average 70-kilogram person, we are talking about a massive 42 liters of water. And here's the number one thing you gotta remember for your test. A full 2/3 of that water is actually locked away inside your cells. That's your intracellular fluid. And the other 1/3? That's the extracellular fluid, basically the sea that all your cells are floating in. The barrier between those two worlds? You guessed it, the cell membrane.
So let's make sure this sticks. The huge majority of your body's water, a whopping 28 liters, is tucked away inside your cells. The rest of it, that extracellular fluid, is then split up again. Most of it is the interstitial fluid that's just bathing your tissues, and a smaller part of it is the plasma that's actually in your blood. Just remember those magic numbers, 2/3 inside, 1/3 outside.
All right, now that we've got the lay of the land, let's zoom in on where the real action is, the cellular frontier. This is where the magic happens, and it's where we're going to tackle the next chunk of our key learning objectives. And here it is, the gatekeeper in all its glory. It's a phospholipid bilayer. You know, you can almost think of it like a tiny sandwich, where the fatty tails are tucked inside and the water-loving heads are on the outside. This makes a perfect barrier. But the real genius here is the proteins that are stuck in it. These guys are the channels and the carriers that give the cell total control over what gets in and what gets kicked out. And that control is the secret sauce that creates those concentration differences that are absolutely essential for life.
Now, what's really cool is how things get across this border. Simple diffusion, that's like an open highway. The more cars there are, the more cars get through. But carrier-mediated transport is different. It has to use a limited number of protein carriers, so it gets saturated. It's kind of like a ferry that only has so many spots. Once it's full, it can't go any faster, no matter how long the line of cars gets. That maximum speed, that's what we call TM.
To really get this to stick, let's use this fantastic analogy. Just imagine that the ions we're talking about are a flock of sheep, and the cell membrane is the fence around their field.
Now, some of the channels in that membrane, the pores, are basically just wide open gates in the fence. They're always open. So our sheep, the ions, can just wander on through, always moving from the crowded field to the less crowded one. They're just following their concentration gradient. Simple as that.
And this just perfectly illustrates the next level of control, gated channels. See, these gates aren't always open. Nope. They need a very specific signal, maybe a change in voltage or some kind of chemical key to unlock them. This is like a shepherd who decides exactly when to let the flock move through. This level of control is absolutely fundamental to how our nerves and muscles work.
So for your exam, you've got to know these 3 heavy hitters. Facilitated diffusion is like the friendly helper. You know, just giving things a little assistance to go down their gradient. But active transport, that's the powerhouse. That's the bouncer at the club, using energy to literally shove things against their gradient, moving them from a low concentration area to a high one.
This is just a perfect example of secondary active transport, and it is such a beautifully efficient system. So the cell spends energy, right? It uses ATP on the sodium-potassium pump to push all this sodium out, creating this really powerful gradient. Then, the SGLT1 transporter uses the energy of that sodium rushing back in to drag glucose along for a free ride, even pulling it against its own gradient. It's like using one powerful waterfall to turn the gears of a whole other machine.
And here's why this isn't just some boring textbook diagram. This mechanism is a literal lifesaver. It is the science behind oral rehydration solutions that have saved millions of lives from diseases like cholera. By giving someone a simple solution of salt and sugar, you kick this co-transporter into high gear. It pulls both of them into the cells, and water just follows along, rehydrating the person and literally pulling them back from the brink. It's incredible.
And now, for the climax of our story. This is the big payoff for all that hard work the cell does separating ions. We're talking about electrical potential. This is the spark of life itself, and it covers our final major set of learning objectives.
Okay, so we've got to get two very important concepts straight in our heads here. A diffusion potential is the voltage that gets created when a charged ion moves across the membrane. Think of it as the little spark generated by that movement. But an equilibrium potential is different. That's the exact voltage you'd need to apply to create a perfect standoff, where the electrical push perfectly balances the chemical pull. So there's no net movement of that ion. It's the voltage of perfect balance.
The Nernst equation is basically your secret weapon for calculating this exact equilibrium voltage for any single ion. You just plug in the concentrations inside and outside the cell, and bam, it tells you the exact voltage that ion wants the membrane to be. For example, potassium's a bit of a homebody. It wants the inside of the cell to be really negative. Sodium, on the other hand, is an adventurer. It wants the inside to be really positive. And all of this careful ion balancing leads to this beautiful dramatic event, the action potential. It is a brief, massive, all-or-nothing electrical signal that cells like your neurons use to basically shout messages across long distances. For your exam, you got to know the key parts of this drama: the stimulus that pushes it to the edge, that lightning-fast spike of depolarization, the dramatic repolarization back down, and then the refractory period, where it kind of catches its breath and resets. So, what's really happening under the hood here? Well, that rapid upstroke, the depolarization, that's caused by a flood of positive sodium ions rushing into the cell. Then, just as the sodium channels slam shut, potassium channels open up, letting positive potassium ions rush out of the cell. And that's what causes the downstroke, the repolarization. It's like a beautifully choreographed dance between these two ions.
Okay, for your exam, you need to have these properties just tattooed on your brain. The all-or-none rule is huge. It means you either get a full-blown action potential or you get nothing at all. There's no in-between. It also travels without losing any strength. And, crucially, those refractory periods act like little traffic cops that make sure the signal can only travel in one direction, which prevents total chaos.
Okay, let's wrap this all up. Here is a quick summary of the absolute most critical, high-yield information you need to have locked in. This is the stuff you should be putting on flashcards, like right now. The source material itself calls this necessary rote learning, so you know it's a big deal. You just have to know this table. I mean, look at those huge differences. Sodium is the life of the party. It's all outside the cell. Potassium is the homebody, mostly wants to stay inside. These two opposing forces are basically the battery that powers everything we've been talking about. They're the yin and yang of cell physiology. And let's end with a final question to really test your understanding. Think about local anesthetics, like the Novocaine you get at the dentist. They work by blocking these exact voltage-gated sodium channels. So, based on everything we've just covered, what would happen to that spark of life if its main fuel source, that flood of sodium, was suddenly cut off? Think about how that stops a pain signal dead in its tracks. You've totally got this. Good luck on your exam.